Ivory Howard of CDRH's Office of Communication and Education. Welcome to the FDA's 54th webinar and a series of virtual town hall meetings to answer technical questions about the development and validation of tests for COVID-19. Today, Dr. Timothy Stemple, Director of the Office of In Vitro Diagnostics and Radiological Health, and the Office of Product Evaluation and Quality, and Toby Lowe, Associate Director for Regulatory Programs, will provide a brief update. Following opening remarks, we will open the line for your questions. Please remember that during this town hall, we're not able to respond to questions about specific submissions that might be under review. Now, please welcome Dr. Timothy Stemple.
Welcome again to this week's call. We look forward to engaging you. We did receive some prior questions, which Toby will go through first. There aren't that many, so we'll go through all of them first and then go to live questions. I did want to make one comment, and that is we still recommend that any molecular comparator for any submission be of high sensitivity with an extraction step, and that it be EUA authorized as used in the sponsor study. If it is not EUA authorized, or if there is an alteration to the EUA authorized method, it will cause a significant delay in our review, because then we'll be required to bring in a molecular review group to look at the method that you use. So if you want to streamline your submission, we recommend you use an EUA authorized test. We also recommend that you check with us first to make sure it's an appropriate molecular comparator. And if there is some requirement for whatever reason for your product to make an alteration to an EUA authorized product, or not use an EUA authorized product, we do recommend you reach out to the FDA and explain that, and let's have a discussion to make sure that that is the most efficient and best way to go forward. That's just gonna help you, the developer group, to have an efficient review by our teams. We have seen issues come up. We've seen issues come up that where sponsors did not do this, in both different cases, and it has caused significant delays in the review. And so we recommend we together try to avoid that. Okay, thanks. Over to Toby.
Thanks, Tim. Thanks, everyone, for joining us again this week. I'll go through the questions that we received through the mailbox prior to today's call. So the first one is about validating changes in the volume of collection media. Specifically, they're noting that they've validated their assays for specific volumes and are considering increasing or decreasing the volume of collection media. And since the LOD is defined in terms of concentration, it doesn't reflect the change in sensitivity of the assay when changing the volume. So we do recommend that you validate your test with the transport media identified in your intended use. Media is provided in a range of volumes, and you should specify in your LOD testing what volume you tested, and note that that's the average or expected fill volume in the VTM provided by the vendor. If you're trying to change the transport media or the volume for your test, we would recommend performing a matrix equivalence study to evaluate the LOD. And you can start with a swab spiked with known concentrations placed in different volumes of the same collection medium, and follow the testing instructions for your test. And that should take into account the dilution factor there. You can also, if your test uses an appropriate extraction step, you could perform a clinical evaluation with samples collected in a larger volume to test the worst case scenario. The next question that we have is asking about pre-submissions for non-COVID projects. As we've discussed previously, the volume of submissions for COVID tests has impacted our non-COVID work. And we did put out some information, I believe it was last week, about through a Voices blog about that workload. And generally, unless an IVD pre-submission is related to COVID-19, companion diagnostics, a breakthrough designation request, or has a significant public health impact, we have been unable to review them. We are using all of the tools at our disposal, and we expect at this point for the remainder of this year to be declining IVD pre-submission requests that don't fall into those categories. Our next question is about a serology assay developer for looking to seek FDA clearance rather than an EUA, and asking whether that would be through a de novo or a 510K. Since we have not yet authorized a serology assay for SARS-CoV-2 outside of the EUA provisions, we would expect that the first submission would be a de novo request. After we get the first one and are able to classify them through the de novo process, so subsequent tests would be able to come through the 510K pathway. Our next question is about the use of serology tests after vaccination, and related to comments that Tim has made in previous town halls about certain tests coming back negative because they don't detect the antibodies that are created by the vaccine. And so this is asking whether FDA's position is that spike S1 antigen-based serology tests are the most appropriate serology tests if a clinician decides to look for immune response from the current vaccine. So we generally don't have formal guidance on this topic. We can, point out that the antibody testing guidelines from the CDC state that antibody testing is not currently recommended to assess for immunity to COVID-19 following COVID-19 vaccination. We don't yet know whether the cutoff for available serology tests would be the same as a protective antibody concentration threshold, since that has not yet been established for SARS-CoV-2. And further studies would be needed to establish a relationship between the detection or a measure of antibodies and protection from infection. So that continues to be a topic of conversation as more scientific and clinical information becomes available.
Thank you, Toby. Yes, this is definitely a very active area of review for the FDA. We simply don't have the data, and we've checked with our federal government partners to support such immunity claims at this moment. As we have authorized for non-COVID, for certain non-COVID infections where monitoring antibodies and antibody levels have been authorized. So there certainly isn't necessarily any prohibition about moving in this area. We simply wait enough data to understand. It's quite likely that a quantitative test is a truly quantitative test may be the best answer ultimately, as we have, as those have been primarily, if not exclusively, the ones that we've authorized for other amenable infections and the vaccinations for them. Okay, Toby, back over to you.
Thanks, Tim. Our next question is about multi-analyte panels, and noting that there is a discussion for multi-analyte panels in the main molecular diagnostic template, but not in the non-laboratory template for home use. And the question is asking whether FDA would consider a home use multiplexed SARS-CoV-2 and influenza test to be eligible for EUA consideration. The sort of overarching answer is yes. The templates are not necessarily meant to be exclusive. There are definitely cases where some of the templates include some crossover where we would expect you to consider whether certain parts of different templates might be applicable to your situation and use the parts that are relevant there. So generally, we are open to multiplexing flu and SARS-CoV-2 in a prescription home use test. At this point, we're not prepared to comment on multiplexing SARS-CoV-2 with other respiratory viruses, but we are open to receiving submissions that propose that approach and having those discussions. We would suggest that you take a look at those templates and complete them as much as possible, with taking the parts that apply, and submit as a pre-EUA for further discussion. We would note that any multiplexed test should be simple and easy to interpret. And as a general recommendation, we suggest that the results not be interpreted via a visual read, but rather that a reader or device to facilitate the correct display and interpretation of the results is used for assays that have more than one analyte. Right. Our next question is asking about the supplemental template for developers of molecular and antigen diagnostic COVID-19 tests for screening with serial testing that we issued back in March, I believe. Specifically asking about if a new, not previously authorized at-home over-the-counter test could be authorized for screening with serial testing based on studies testing only symptomatic individuals, as long as there's a post-authorization commitment to provide data from testing asymptomatic individuals. Yes, the answer is yes. That is the intent of that template is to leverage the strong performance in symptomatic individual to authorize for asymptomatic screening. So if all of the other requirements or recommendations are met, including especially for at-home over-the-counter, including simple to use, appropriate way labeling, and validation with way users, we would consider that that asymptomatic screening claim with a post-authorization condition of authorization in the letter of authorization for the developer to conduct a study to establish performance with asymptomatic individuals. Our next question is about a prospective clinical study supporting 510K submission of a SARS-CoV-2, influenza, and RSV multi-analyte molecular RT-PCR test, asking whether FDA has any expectation on the percentage of specimens tested fresh versus frozen, and if using frozen specimens, asking about fresh versus frozen and frozen stability studies, asking about the requirements for that, for frozen specimen stability. So to support a 510K, we would recommend that your prospective clinical study not exceed 50% frozen prospective specimens. We typically recommend that sponsors retest any frozen specimens with the comparator method prior to testing with the candidate method. And a fresh versus frozen study would establish that the test has similar performance with both sample types. And we've mentioned previously in these town halls as well that if you're intending to pursue a 510K pathway, we recommend that you submit a pre-submission to discuss your clinical validation for review.
Thank you, Toby. Any comments?
Great. And our last question on the ones that were sent in ahead of time is about at-home, over-the-counter lateral flow neutralizing antibody assay using a finger stick, asking about clarifications for the study design, for clinical agreement and matrix equivalency studies, whether the clinical agreement, for the clinical agreement study protocol, asking whether they need to run both serum obtained from venipuncture whole blood as well as finger prick whole blood against the gold standard comparator, PRNT. First, we would note that we would want to have further discussions about this home use test, and particularly whether prescription home use or over-the-counter home use would be appropriate. So we would recommend you coming in to talk about that with us. For the clinical agreement study, we would recommend that you evaluate, finger stick samples in a clinical agreement study, and that you collect paired venipuncture samples from subjects so that you're able to run the neutralization comparator method. Finger stick whole blood claims should be evaluated in the clinical agreement and not in a matrix equivalency study. And that is consistent with the recommendations in the serology template regarding finger stick evaluations.
Thank you, Toby. I think that's the last submitted question, correct?
Yes, it is.
And so I think we can go open up the line and take live questions. Thank you, Toby. Well done.
As a reminder, if you would like to ask a question over the phone, please press star then 1. Record your name clearly when prompted. Be sure that your phone is not muted when you're recording your name. And our first question comes from Richard Montagna. Your line is now open.
Yeah, thank you. This is Richard Montagna from Rionics. I'm sure all the other authorized manufacturers, we've been continually monitoring these various emerging variants. Initially, we were using in silico analysis to establish whether our test would pick up these variants. And recently, we've managed to get our hands on some actual variants that we could do wet lab testing. So based on that, I have two questions. The first one is, and I'm sure everyone else is getting these same questions from their customers as to how our tests would stack up against these emerging variants. Assuming we were able to provide to FDA in the form of an amendment to our EUA the data that we've accumulated, would FDA permit labeling changes to allow us to make those claims? And a second related question is, has FDA considered, or would FDA consider providing a blinded panel of samples that would contain the various variants of concern at various titers, so that people could compare head-to-head in a very unbiased head-to-head manner the relative performance of these various tests? Thanks.
Thank you, Richard. Interesting question. I think we'll take that back for a dialogue within the FDA. We're not, unfortunately, we're still staying incredibly busy, and we're not looking for additional work to do. On the other hand, variants and mutations are extremely important. We have been updating labeling when there is a potential issue, and if there's data around a potential issue, just the magnitude of that. We've got guidance out there. We've also got a new website. And our pledge to the community is that As soon as we know something that could be an issue of significance in the United States, we will reach out to the developer or developers and assess that. We're also looking ahead to variants that are either in extremely low abundance in the U.S. or not here yet, but are present in other countries. So we are looking at in the important variants in other countries that may in fact spread to the U.S. and become dominant enough to affect test performance if there's an issue. I understand your desire here. Probably, and I'm not. This is another definitive answer, but probably we'll want to restrict our activities to addressing potential issues with specific tests. Part of this is we don't want to make work for developers either. And we certainly remain swamped with work at the FDA. Not just COVID-related work, but non-COVID work as well. So hopefully your understanding of a potential, response here that may come next week on this town hall to your question.
Well, thank you very much. Appreciate it.
Thank you. And our next question comes from Elizabeth Brunelli. Your line is now open.
Hi, this is Elizabeth Brunelli from Ipsum Diagnostics. For the purpose, and this is a three-part question. So for the purposes of the home collection kit EUA request, we noticed that the template includes entries for both a proprietary and established name. What is the difference between the two?
Toby, do you think you can respond to this question?
I'm trying to pull up the template right now to see. I think that is, generally referring to if you have a specific name for your device, and there is also a more generic name for it. But why don't you go ahead with the rest of your question, and I'll pull that up and take a look.
Okay. This is just really quick on the oral swabs that are used in the home collection kit. Does the brand of oral swab need to be specified at this point? And if so, is FDA registration of the swab necessary for each type of swab you might use?
I was thinking Toby might be answering that. Can you repeat that question? Sorry.
Does the brand of oral swab need to be specified on a home collection kit EUA request? And if so, is FDA registration of the swab necessary or required?
It's preferred that be registered. If it's not, I would have dialogue with our staff. In the submission, I would state all the details of the swab used in the study. Now, when it comes down to it, if there are a need for a developer pre-authorization to switch swabs, would want to know about it in our review. But also post-authorization, in order to keep supplies on market, we're going to, expect that if it's otherwise the same exact swab, i.e. the same material, the same type of swab, that validation can be done internal to the developer, and as long as everything looks good, it does not require a review by the FDA. And I'm not sure if Toby has gone to the template and been able to address your question yet. Toby?
Okay, so what I'm getting from that is that initially on the EUA, we should use the FDA registration for the original swab. If we need to substitute that, then we can do probably a validation in-house for the new swab, as long as it's the same material. Correct?
Yes, yes, and at this point, we're not hearing about wide-scale swab shortages. So we would prefer to use an FDA registered swab. If you're not, I think that's the time. If you need to switch to something that's not FDA registered due to supply issues, I would request that you reach out to the FDA and get some input on that before you do the work.
Okay.
And I think perhaps we can find an alternate source that's FDA registered for you. Go ahead, Toby.
Okay.
Yeah, I think, we also, since the home collection kit is something that you will be distributing, we would want to know specifically what the components are in there. And so we would want to see, we would want to know the brand of the swab and, especially if it's not something that you intend to manufacture yourself, we would want to know, to make sure that it is a registered and listed device so that, if there was a recall or anything, we know who's responsible for that.
Okay.
And for the question about the proprietary name versus the established name, I can try and get some more information for that to get back to you on, but I believe that it's referring to the legal name of the device, the proprietary name from your company, and the established name would be sort of if there's a more generic common name that you use for the device.
Okay, and if you don't have one, you don't need to include it?
Yeah, you can use the same thing for both if that's
Okay.
the situation for your test, for your device.
Okay.
Yep.
And the last part is, in the non-prescription home collection molecular PCR model combination, can two separate EUA request documents be submitted at once, or do they need to be combined into a single document?
Sorry, can you say that again? For what type of
Yeah. I didn't
device?
catch it either.
Non-prescription home collection combined with molecular PCR at the laboratory. There are obviously two separate EUA requests. Can those be submitted together in one email, or do they need to be combined into a single document?
So they can be separate or together in terms of a single EUA versus two EUA requests, depending on your situation. So if you have a home collection kit that you intend to only use with one single assay, and they're both under the same EUA sponsor, you can submit that as a single EUA request for the system as a whole, the home collection plus the assay. If you instead intend for the home collection kit to be used with multiple assays, and or you intend for the assay to be able to use multiple home collection kits, then we would want to see two EUA requests so that they can be labeled appropriately. And in that case, you could submit them in one email or two, although it is probably a little bit cleaner if you submit them in two emails.
Yeah, and if it's different developers for the collection kit and the central lab test, then it will be two EUAs or two amendments, and it's probably best to keep them separate. So we can clearly delineate each submission in our record keeping. And we're going to need to move on to the next caller. Thank you.
And as a reminder, if you would like to ask a question over the phone, please press star one. Please limit all of your comments and questions to one comment and question per caller. One moment as we wait for the next question.
You may be muted.
And one moment for our next question. our next question comes from Aaron Erickson. Your line is now open.
Hello, this is Ariane Erickson. I'm from Zenith Health. So I have a question regarding point of care antigen IVD. So for the evaluation for EUA submission for a point of care antigen IVD, can all aspects of patient sampling and operation for our investigational device be conducted by the typical point of care personnel? So this would be non-laboratory personnel and medical personnel. But we would like to conduct this study on CLIA site, but in a separate demarcated area where our personnel would have no contact with the laboratory personnel.
You were breaking up a little bit, at least for me. You have a point of care antigen test that you want to evaluate, and typically we do recommend that it be performed in actual CLIA waived settings in the U.S. With typically non-laboratorians. Obviously, these are healthcare workers, but not trained laboratorians, in order to evaluate the performance. But there was a twist I think you introduced tonight, and you're breaking up, so I didn't understand the twist.
Sorry, I apologize. Can you hear me now?
Yeah.
Perfect. So we're looking to do the testing in basically a simulated point of care environment. This would be because site where we're running PCR is a CLIA site. We would like to do it in a similar location for sample collection, use a sample collection for our operators.
We continue to recommend that you use typical point of care sites. You can also use, there's a whole lot more CLIA waive certificate labs out there, including at schools and workplaces. The reason to recommend that these types of settings be used is if they're doing this kind of work, they're a busy situation, and they're running they're doing lots of things all at once. In a typical point of care clinic, they're seeing patients for a lot of other issues other than COVID. And they then intersperse work for you or a point of care device that was already authorized in all their other work. So we want to see in a busy clinic situation, for example, that they still can follow the directions, it's robust enough that they can get the accurate results. So simulated situations for point of care are not something that we would recommend. And if you absolutely have to go that route, and I can't dissuade you on this call, then I recommend you reach out to our FDA staff and discuss this with us. We are allowing simulated home environments. That's a different situation. Because a home user they're dedicating their attention to running this test. And rather than require developers to use it in home, we're making it easier to get access to home testing. We're bringing those home users into a simulated home environment, but they get no assistance. They're handed the kit, and that's it. There's no training or anything. So hopefully I addressed your question.
And our next question comes from Laura D'Angelo. Your line is now open.
Great. Thank you. Hi, Tim and Toby. I was hoping to follow up on something that we talked about briefly last week, which is difficulty accessing BSL-3 sites. And I think Dr. Stenzel, you mentioned that there are some workarounds, and I did not write them down. So could you just briefly go over those again quickly?
Yeah, and I just want to remind everybody that the transcripts from these calls are posted. Toby, do you know if the transcript from last week has already been posted? So I'm not sure if I'm going to go through all of this quickly, but I'll just go through it quickly because there may be new callers on the line today. But yeah, we do not require BSL-3 facilities for any test validation. It's only if a developer wants to use live virus that they have to do that. But There are inactivated viruses. They're heat inactivated. There's these are for analytical studies, not for clinical studies. Heat inactivated or irradiated virus that can be obtained from VEI and potentially other sources. And then actual patient, residual patient samples can be used. For analytical studies, sometimes you can dilute that down in negative patient matrix to an appropriate level for the given analytical study. So again, BSL-3 level labs are not required to be used by the FDA for any test validation. And it's only the developer at this point who may want to use a live virus that would do that, but that would be on their own accord, or anything that's specific for their device that requires that. And then please do check my comments on the transcript that are posted for last week. Thank you.
Thank you.
And just to clarify, last week I don't believe it's posted yet, but it should be posted very shortly.
All right. And the next question, I believe the caller's name is Whitney Gell. Your line is now open.
Hello? Is that me?
Hello?
Your line is open.
Hello? Is that from me? Whitney
We can hear you. Go ahead.
Yes. Okay. Yeah, I have a question regarding the home collection kit for multiple molecular acids. So, must all those acids be EUA? Can the home collection kit be used for LDT as long as it has the RP in there?
So tests that are used for home collection do need to be authorized. So we would expect that the both the assay and the home collection kit be authorized and be authorized specifically for that indication.
I see. So they have to. Okay. So If for LDT, how we can, if we want to use the authorized home collection kit with LDT assay, how should we follow? Should we follow the clear regulation, like CMS, or we cannot even do that? Or
I'm not quite sure I'm following. The test would need to be authorized through an EUA, so we would expect an EUA request to be submitted to FDA.
If there are kit developers out there who are thinking about, having a lab use it as LDT, but they've provided a kit with complete instructions and it's built for use for clinical purposes, that would not be a good thing to do. And we don't recommend that. And if we do find a kit manufacturer who's doing that, it would be important for us to reach out to them and at least have a discussion about what's going on. So, and that's intended for anything intended for clinical purposes. So, it's not a recommended way to get into the United States.
Right, and to clarify, a test that is intended to be used at a clinical lab, a test kit that is intended to be used at a clinical lab for clinical testing, does not meet the definition of RUO, or research use only. So there should not be RUO test kits being sent to clinical labs for clinical testing, to be quote unquote called an LDT.
Yeah, and I'll just say I signed another warning letter yesterday about the marketing of an unauthorized test in the United States. So kit. So this is something that the FDA takes a serious look at. All right. If there's another caller, we'll take it.
And as a reminder, to ask a question over the phone, please press *1. Our next question comes from Anna Powell. Your line is now open.
Hi, this is Anna Powell. I have a question regarding rabbit antigen POC EUA submission. So far, we have conducted a clinical study with prospective samples. We have very good performance but only have 22 symptomatic and 10 asymptomatic. Now, we know that the EUA template requires the 30 positives and more. I'm talking about positive sample here. Would the 10 asymptomatic be sufficient to make the total number more than 30? Or you need us to obtain additional symptomatic positive sample?
That's an interesting question. I think typically we want to see a full set of symptomatic samples so that we can see, is this a molecular test or an antigen test?
Our headline was removed. Please press star 1 again, and I'll reopen your line.
It was antigen, Tim. She said it was a rapid antigen point of care.
Yeah, the challenge with an antigen test is we really do want to record the days since symptom onset. So we know how many days that a test is going to be performing well, and so purchasers and users of that test know the window after symptoms for symptomatic patients in which the test performs. So if you include non-symptomatic patients who legitimately can be COVID positive, we're not going to collect that information. And with only 20 or 22 cases, our confidence intervals around the sensitivity and the days after symptom onset that it is sensitive enough for authorization are more limited. So fortunately, it is easier to enroll symptomatic patients in clinical studies and identify them and get them and get positives that way than it is asymptomatic. The fact that you had 10 asymptomatics and perhaps performance is good in them is encouraging because it means that once you get those additional symptomatic subjects enrolled and your performance remains good in the symptomatic population. When you submit and the data all looks good, you have the opportunity, if you wish, to apply not only for symptomatic claims but also asymptomatic screening.
Thank you. And our next question comes from Alexis Powerbench. Your line is now open.
Hi, this is Alexis Powerbench from Exponent. I had a question with regards to a study design for a breath analyzer, which is based on antigen detection. Would an appropriate comparator still be recommended as a high sensitivity molecular test, or should we also consider antigen tests as a comparator?
Antigens are typically not a good comparator because they're not as sensitive as high sensitivity molecular central lab tests with an extraction step. You have a breath test that looks for antigen?
That's right. Mm-hmm.
Okay. Well, the comparator would still be at least a... actually, I don't know what the true comparator is here, but what swab type it is. But if you email our template email address, we can give you some feedback for a breath test recommendations. We're still working on a template for these sorts of tests, and we'll post them as soon as we can. But we can provide some feedback now if you email our template email address.
Yeah, thanks. We actually already did that, and we're working on a pre-EUA submission to have a further discussion, but just wanted to get some early feedback. Thank you very much.
Uh-huh.
Thank you. As a reminder, to ask a question, please press star 1. Our next question comes from Albie Ling. Your line is now open.
Hi, my name is Albie Ling, and I have a question about the clinical evaluation study for a molecular test we're developing. So it's been really difficult to recruit COVID positive patients, and we're just wondering, what if we have some intended users run the test on COVID positive, like the saliva samples collected elsewhere? Is that a, okay study design given the limitations these days? And if that's okay, would you prefer to have, let's say, 30 intended users perform a test on each of the saliva samples we have collected elsewhere? Or would it be okay to have, let's say, 5 intended users to perform all 30 tests on the saliva samples we have so far?
Tim, are you taking this one? You might be on mute if you're talking.
I was talking away. I have some of the best conversations with myself that way. So molecular test targeting detection of virus in saliva. Is this a point of care test?
It's going to be a take-home test that's going to be over-the-counter use.
Okay. So I recommend you come in with a pre-EUA requesting this. That's going to be really, really challenging for a new test that hasn't even received authorization, say, for a point of care or for central lab environment. If you have a central lab test, moderate or high complexity, you can use banked samples for a molecular test, but they do need a matched banked saliva samples. But you need a matched NP swab for that sample, and you do need to also have some fresh samples, fresh positives. But this is going to be extremely challenging. We understand that viral positivity rates in the United States, fortunately, are going down. Hopefully they stay down. And then there's a challenge for new test developers coming into the market now, and especially with challenging substrates like saliva to get positives. So, I don't think our recommendations for validation are going to change yet at this time. But to do reach out to our staff through the template email address, or primarily, I think, through a pre-EUA. And if you want to suggest alternatives, we will review them.
Thank you so much. Can I ask one additional question, or does everyone only get to ask one?
Go ahead. Hopefully it's quick.
Right. So that was about clinical evaluation. So this is about usability. Does FDA require or recommend us to have the usability study conducted on positive patients as well? So it's not really clear from the template.
So usability study is more making sure that home users, in this case, can follow the directions and perform the test accurately. So we have potential alternatives there to actually having positive patients. So again, I would propose something in a pre-EUA and send it to our team.
All right. Thank you so much.
As a reminder, to ask a question, please press star 1. One moment as we wait for any additional questions. Not showing any additional questions at this time.
We can hold on for a minute or two, see if there's anybody else that wants to ask a question. If not, we can close the call.
Absolutely. As a reminder, to ask a question, please press star 1. One moment as we wait for any additional questions.
Okay. I don't think we're going to get any other questions, so we can turn it back over to Ivory for closeout. Hello, Ivory?
Okay, I'm here. Thank you. This is Ivory Howard. We appreciate your participation and thoughtful questions during today's town hall. Today's presentation and transcript will be available on the CDRH Learn webpage at www.fda.gov/training/CDRH-Learn. If you have any additional questions about today's presentation, please email cdrh-eua-templates@fda.hhs.gov. As we continue to hold these virtual town halls, we appreciate your feedback. Following the conclusion of this webinar, please complete a short 13-question survey about your experience. The survey link can be found now in the chat box or online at www.fda.gov/CDRH webinar. Thank you again for participating. This concludes today's virtual town hall.